Okay, so we're starting off with sort of a general overview of the acid base and what you can expect, what's important, and that's really the compensation. So being able to identify a disturbance and identify the compensation. So these last 4 slides are going to be a really good place to focus your studying, as well as this flow chart here at the end. And so we talked about in the lecture being able to differentiate the cause of an acid-base disturbance. So we know that an acid-base disturbance is a pH outside of our normal range, so outside of 7.35 or 7.45 on the upper end of that scale. So that is just the disturbance itself, right? It can be an acidosis or an alkalosis. But then we really want to hone in on the cause, okay? And so we're going to look at some other parameters to help us do that. Sorry, I'm flip-flopping here. So here, we'll start with respiratory acidosis. We know that this is a lower end of the pH, so anything below 7.35. And so the cause, because this is respiratory, the cause is going to be a change in ventilation, so a hypoventilation scenario. It can also be an increase in acid that is related to CO2, right? So CO2 poisoning, anything that is an acidosis, but is caused by a change in ventilation or an increase in CO2. So if we have an ingestion of acid, that's not going to be a respiratory cause. That's going to be a metabolic cause, okay? And then also thinking about the compensation. If the cause is in the lungs or the respiratory system, the compensation is going to be in the kidneys, right? So if we're having a change of ventilation, the kidneys are going to be the system that help to compensate. So we're going to see them increase their acid secretion, so push more acid into the filtrate to be excreted from the body, or increase their bicarbonate reabsorption, so bring back more of that bicarb from the filtrate into the blood to correct that acidosis. So we want to be thinking about, in the big picture, that if I have too much acid, that can be a problem. But if I lose too much acid, bicarbonate will take over. That can be a problem as well. So we want to keep these two elements balanced. And so losing or increasing either of those is what is going to give us these initial disturbances. Going on to respiratory alkalosis here. So the cause here again is going to be a respiratory cause. So hyperventilation in this scenario, or a decrease in CO2, which is resulting in a decrease in acid. So in the video, I would have talked about the mechanism by which CO2 is converted to acid with carbonic anhydrase, carbonic acid. And so if we have decreased CO2, we have decreased acids likewise, right? If you're hyperventilating, you're blowing more of that CO2 off more quickly, you're going to throw yourself into an alkalosis because you're losing too much acid. And again, we're going to compensate with the opposite system. So the kidneys are going to kick in and try to compensate for that initial respiratory disturbance by decreasing their hydrogen ion secretion or decreasing their bicarbonate reabsorption. So if we have too much bicarbonate in the blood because we're losing too much acid, we either want to stop secreting as much acid to help balance that bicarb, or we want to stop reabsorbing that bicarb to help, again, balance that scenario. Metabolic alkalosis, so the metabolic disturbances are going to be caused anywhere outside of the lungs. So we're going to think about ingesting something that can be acidic or alkaline. We're going to think about things like vomiting. If you vomit a lot of acid in the stomach, you lose that acid, so that's going to be an alkalosis. Or we can think about diarrhea. When you have diarrhea, you lose bicarbonate. There's a lot of bicarbonate in the colon. You lose that bicarbonate, and that can throw you into an acidosis. Okay, also think about things in the diet. So in the lecture, I would have talked about protein. High protein in the diet breaks down to hydrogen ions, which is acidic. High fat in the diet breaks down to hydrogen ions. That's acidic. If you're doing heavy lifting at the gym, right, we know that the muscles generate lactic acid, especially those glycolytic muscles. And so that also can contribute to acidosis. So anything happening outside of the lungs is going to give us these metabolic disturbances. So metabolic acidosis, if we have an increase in hydrogen ions, independent of CO2, in other words, independent of the respiratory system, and the compensation is going to happen in the kidneys. So if we have a metabolic disturbance, the kidneys can kick in, they can change the ventilation, right? They can increase ventilation, which is blowing off more of that acid via CO2. The kidneys can also compensate, but it's least likely to see the kidneys compensate for a kidney disorder. So if it's a kidney problem, the kidneys are not going to compensate. The lungs are what are going to compensate. But if it's not a kidney problem, it's something you ingested in the diet, or again, lifting heavy at the gym, the kidneys can compensate as well. So they can increase their hydrogen ion secretion to kick more of that acid out. They can increase their bicarb reabsorption to bring more alkaline, bring more bicarb to balance that acid out, or they can make new bicarb by the proximal tubule. And then lastly here, metabolic alkalosis. So again, just the same idea here where there's not a problem with the lungs, it's some other cause of this disturbance. This one is an alkalosis. So this is the one we're thinking about diarrhea, where we lose all that, vomiting, excuse me. So diarrhea is going to be an acidosis. Vomiting is going to be an alkalosis. So we're losing all of that acid when we vomit. And that is going to throw us into a metabolic alkalosis. So anytime we lose hydrogen ions that is not associated with CO2, so not associated with ventilation, that can initiate a metabolic alkalosis. And the compensation is going to be in the respiratory system. If it's a kidney problem, so if the kidneys are malfunctioning and they are not secreting enough hydrogen ions, or they're secreting too much rather, then we're not going to have the kidneys compensate because the problem is in the kidneys. The lungs are going to compensate. But if the kidneys are not the main problem, such as vomiting, right, the kidneys can also kick in and they can help to compensate that by decreasing the hydrogen ion secretion. So losing less acid, right? We want to hold on to more of that acid if we have an alkalosis. They can also decrease their reabsorption of bicarb, so reabsorb less of that alkaline bicarb, and then they can also decrease the synthesis of new bicarbonate. All right, so these are the four main slides you want to go back over and think about some practical examples of the causes, and think about where they fit. Also think about what the compensation is going to look like, right? As far as this flow chart, I don't expect you to commit this to memory, but I do expect you to have an understanding of these parameters. So let's say, expect that I can tell you a patient has a pH of 7.5, right? Which is high on the scale. If I tell you that they had a low PCO2, you should be thinking about a respiratory alkalosis. If I tell you they had a normal PCO2, but a high bicarb, you should be thinking about a metabolic alkalosis. So not necessarily committing these to memory, but understanding the fluctuations, right? If the PCO2 is normal, we're not looking at the respiratory system on either of these, whether it's acidosis or alkalosis. If I have a normal PCO2, I'm not thinking about a respiratory disturbance. If I have a normal bicarb, but I have an abnormal PCO2, I am thinking about a respiratory cause, okay? So be familiar, be comfortable applying the information on this table to a practical scenario or a patient, right? If you're given the pH and then you're given the parameters of PCO2 and of bicarb. All right, any questions remaining about acid-base balance? Any questions there? Okay, probably at the end, we'll try to go over some of these practice questions. I thought they would be helpful, but I don't know how much time we'll have. So I'm gonna move right along, excuse me. I'm gonna move right along to sort of an overview here. And again, in the interest of time, I took some time to actually craft this overview. It usually takes me so much time to write this up. So I'm going to go over this, but I'm also going to provide this drawing as a part of the recording today if you'd like to go over it. And so, let's see here. Okay. So this is an overview of the kidneys, of the different parts of the nephron specifically, and of the important processes and features that were found in each part. And at the beginning, I kind of asked you to start crafting this overview as a good way to plug the important details in each area. So we want to think about the glomerulus here, filtration happening here. And so I haven't filled in the exchange processes, but we know that filtration is occurring here across the glomerulus. And so we want to think about these important factors about filtration. So we've got the cortex. This is where this part of the nephron is, whereas these other parts are down in the medulla, right? This part, sorry, excuse me. This part is down in the medulla. We want to think about the Starling's forces, and I'm going to go over those in detail coming up. So the 4 Starling forces that are acting across this filtration membrane. We also want to think about the filtration membrane, right? So what are the structures that the filtrate has to go across? That's going to be the endothelial cell of the glomerulus, the basement membrane, and then the epithelial cell of the Bowman's capsule. Okay, so although I haven't written that out here, we want to also think about the filtration membrane, right? Other important details in this region are going to be the intrinsic regulation. So the factors that help to keep GFR at 125 in the face of a changing mean arterial pressure. So if mean arterial pressure is between 80 and 180, GFR stays at 125. What are those 3 intrinsic mechanisms? So we had myogenic feedback, we had the tubulo-glomerular feedback, we had the mesangial cells, okay? So the myogenic feedback were the afferent arterial cells, so essentially the JG cells, okay? Tubulo-glomerular feedback were the macula densa cells, and then the mesangial cells were the ones that were kind of stuffed in between here, right? And I'll talk about those cells coming up because someone did ask me to go over the JG complex, and so I'll touch on those again coming up. Also, we want to think about the extrinsic regulation. So the sympathetic nervous system acting here as well. So if mean arterial pressure is less than 80 or greater than 180, then GFR will rise or fall accordingly because of the sympathetic nervous system, which is an extrinsic regulation onto GFR. So this is not within the kidneys. This is outside of the kidneys by the way of the autonomic nervous system. This then brings us to the proximal convoluted tubule. So the important features here were the leaky tight junctions. This allowed for some paracellular transport. So the cells here are adapted for reabsorption. We see mass reabsorption happening here, especially of water. We also talked about the brush border. So the cells here have those apical modifications, which throws the epithelial layer into these folds that helps to increase the surface area, and that is known as the brush border. Here we see unregulated reabsorption, so no hormones, no autonomic nervous system, nothing external or extrinsic is impacting the reabsorption that happens across the proximal tubule. As far as what is reabsorbed, we talked about most of the things being reabsorbed: amino acids, nutrients, glucose, our ions, sodium, potassium, chloride, et cetera, bicarbonate as a way of balancing the acid base we just talked about. And those things are going from the epithelial epithelial lining, so through the epithelial cell of the proximal tubule, across the interstitial fluid, and into the endothelial lining of the peritubular capillary. So have an understanding that wrapping around this area is the peritubular capillaries. Okay, our peritubular capillaries. Also wrapping around the distal tubule. So these are also our peritubular capillaries. And that is how we facilitate the second renal exchange process, which is, reabsorption. Okay, moving things from the filtrate back into the blood via reabsorption. I didn't write it out here in the interest of space, but know the mechanisms, okay? Know these mechanisms. Solute reabsorption, which facilitates water reabsorption. Solute reabsorption is happening by secondary active transport across the apical surface of those cells. And then it's happening by the sodium-potassium pump, which is primary active transport, and facilitated diffusion across the basolateral membrane of the cells. So make sure you get in here and review the mechanisms by which solutes, and water are reabsorbed, okay? Moving on to the loop of Henle, this is where we encountered 2 important phenomena. So the medullary osmotic gradient, this idea that down here in the medulla, we have a higher osmolarity, right? It's more salty, it's more concentrated. Up in the cortex, we have a lower osmolarity, right? So we've got less solutes and we have more fluid, more water here, okay? And the reason for that is because we have solutes being actively pumped across this end, right? So we have sodium, chloride, potassium being pumped out here, whereas we have water being pumped out this side, okay? So only permeable to water down the descending limb, only permeable to solutes up the ascending limb. And so we talked about how we establish that. We push some fluid down, we recalibrate, pumping solutes out, water out, push some more fluid down, pumping solutes out, water out, push some more fluid down, until we get to a steady state where we have the deepest depths of the medulla having an osmolarity of 1200 to 1400, whereas up in the cortex, we have an osmolarity of 100 at the end here, whereas we started with an osmolarity of 300 over here. Okay, so we started with an isoosmotic scenario, and then we end up with a gradient at this end because of that interstitial fluid gradient going all the way down to the medulla. So that was the counter current multiplier. We also spoke about this idea that urea, really important. I'm going to go over this at the end again, but just to touch on it here, urea is what maintains the gradient. Okay, we also introduced this concept that the vasa recta, which is coming into contact with the loop of Henle, is running in the opposite orientation. So the vasa, so this is the descending limb. Here's the descending limb of the loop. Here's the ascending limb of the loop. The vasa recta runs in the opposite orientation. So here's the vasa recta. Okay, it's going down in concert with the ascending limb of the loop, and then it's going up in concert with the descending limb of the loop. So the solutes that are being pumped out here are actually going into the vasa recta here, along with urea. So on this limb of the vasa recta, we have these UTB receptors. So urea is also going into the vasa recta here. And that is preventing us from losing the gradient that we just worked so hard to establish. So we don't want those solutes to linger here in the interstitial fluid. We want them to go out of the loop, but we want them to go into the blood. So we have the solutes being moved in here, but also urea via the UTB channels, okay? On the ascending limb, we don't have any urea channels because now we're moving all of that water out. So we now have the gradient by which water, we're moving that water in. So we now have the gradient by which water can move into our filtrate because it has a higher osmolarity. We just pumped all these solutes in on this side. So the osmolarity of the blood here is really high. And so now we have a gradient for the water that's leaving the descending limb of the loop to go right into the ascending limb of the vasa recta because we just pumped urea and all those solutes in to create that higher osmolarity. So now we have a gradient for osmosis on the ascending limb of the vasa recta so that water can just rush in. And the whole role here of the vasa recta and this movement of the things from the tubule into the blood is so that they are not staying in the interstitial fluid that would dissipate the gradient that we just worked really hard to create. And so those things that are moving from the tubule, so solutes on this side, water on this side, have to move into the vasa recta, solutes and urea on this side, and water on this side. Okay, and so essentially the osmolarity of our blood stays the same. We have an osmolarity of our blood starting at 300 here. We add solutes and urea, so it becomes really salty down here, lots of solutes, and then we add water going up here, so it gets back to 300. Okay, and that is how we can reclaim those things that we're taking from the tubule, from the tubular filtrate, without necessarily manipulating the osmolarity of our blood. It stays the same. Okay. All right, any questions on that bit? Any questions there? Okay, and if you have questions as I'm going through, you can feel free to drop them in the chat. Let's move on to the distal tubule and the collecting duct. Here is where we see reabsorption, but we also introduce another exchange process, which is secretion. So we have reabsorption happening here, but we also see a little bit of secretion happening here as well, much more than we saw at the proximal end. The reabsorption here, however, is regulated. Okay, and I'll touch on what that means here again in a bit. Here, it was unregulated. I apologize if I'm reiterating information that's already written. It may make this a bit jumbled, but just to reiterate, reabsorption here is regulated, means that it's manipulated by hormones. And so we really want to talk about the hormones as far as looking at what's important here in the collecting duct. First of all, we want to say that the structures here are different. So we have tight junctions, right? So the cells here don't allow for that paracellular transport the way that we see here with the leaky tight junctions. We also don't have microvilli, we don't have the brush border, we don't have that free reabsorption, that adapted membrane that is suited for unregulated reabsorption the way that we have here. The reabsorption here is manipulated and controlled by the hormones that we're going to speak about. So the hormones are ADH, so antidiuretic hormone, or vasopressin. This is going to insert more aquaporin 2 channels into the apical surface, as well as more sodium-potassium pumps. Okay, excuse me. I'm jumping ahead of myself here. So we're inserting more aquaporin 2 channels into the apical surface of the principal cells here. So it's probably not written here, but we are talking about the principal cells. Okay, at the collecting duct and distal tubule. And so ADH coming from the posterior pituitary will insert more of these aquaporin 2 channels so we can reabsorb more water. I'm going to talk about the stimuli for ADH in another slide coming up. Aldosterone is another hormone here. This is going to increase sodium reabsorption. It's going to do two things. It's going to insert more sodium-potassium pumps on the basolateral membrane. It's also going to insert more leak channels for sodium and potassium on the apical membrane. Okay, so sodium and potassium, right? Sodium is going to facilitate inward movement. Those leak channels facilitate inward movement. Potassium channels are going to help with potassium secretion. So they're going to facilitate more outward movement of that potassium. Okay, and all of that is manipulated by aldosterone. Again, I'm going to touch on the stimuli for aldosterone coming up. The third hormone here is ANP, atrial natriuretic peptide. That is going to decrease sodium reabsorption. So this is the antagonistic hormone of aldosterone. We're going to waste more potassium here, lose it into the urine because we're going to decrease the reabsorption, and that is being done by blocking the leak channels on the apical membrane. Okay. All right, any questions on this sort of general overview of kind of all the processes and features of the nephron? All right, and again, I'm going to save this. So if you missed anything, this will be with the recordings today. And so the blood supply, really just understanding these branches of the renal artery and where they're located. So we kind of just segmentally branch from the renal artery. We then go into segmental arteries, like so major branches here, these larger chunks are the segmental arteries. Then we have the interlobar arteries. The interlobar arteries are actually running in the renal columns. So remember, this part of the kidney is called the renal column, in between the renal pyramids. We then get into the arcuate arteries, so the ones that kind of loop over the base of the renal pyramid, right here. So these are called arcuate because they're kind of bending over the base here. And then we branch into our cortical radiate arteries. So these fine branches that we're seeing here right out in the cortex, these are the cortical radiate arteries. These are what give rise to the afferent arteriole. So the afferent arteriole will then feed into the glomerulus, right? So the glomeruli are going to be out here in the cortex, and so that makes sense, right? So we go from the segmental, these large branches, to the interlobar going in between the pyramids in the renal column here, to the arcuate branching or bending around the base of the renal pyramid, to the cortical radiate, the very tiny branches here, to the afferent arteriole, to the glomerulus. Okay, and then we branch in the opposite direction for our veins. So the veins, the corresponding veins for the cortical radiate arteries are actually the interlobular veins. All right, so we don't have an interlobular artery. The interlobular veins are corresponding with the cortical radiate arteries. So they're kind of in the same place here. And they're coming from the glomerulus, which is essentially coming from the vessels that are wrapping around the nephron. So we have the peritubular capillaries, the vasa recta coming into contact with various parts of the nephron, right? So the vasa recta coming into contact with the loop, the peritubular capillaries coming into contact with the proximal convoluted tubule and the distal convoluted tubule, and that is coming from the efferent arterial. So after we filter that blood across the glomerulus, we then go into the efferent arterial, and then we go into these structures that are wrapping around the rest of the tubular structures so that we can capture the things that we didn't want to filter out and pull them back into the blood this way. And then we go into the interlobular veins, or the tiniest veins out here in the cortex. Then we sort of congregate into the arcuate veins wrapping around the base of the renal pyramids. They are corresponding with the arcuate arteries. Then we get into the interlobar veins. They are corresponding with the interlobar arteries within the renal column. Then we get into the renal vein, and then we get into the inferior vena cava, okay? So that is the blood supply going out all the way to the nephron and coming right back to the renal artery. Or sorry, the inferior vena cava, sorry. Okay, so pretty straightforward, that's the blood supply. Any questions on blood supply? Any lingering questions on blood supply? We're gonna move along to GFR. So let's go over GFR here. I think it was in this PowerPoint. So please be familiar, be comfortable, brush up on your Starling forces. Make sure you know which ones are favoring filtration. So at the beginning of your exam, you can write out this key on a sheet of paper. This is gonna help, it's gonna go a long way. Know which forces favor filtration and which forces oppose filtration, right? So the forces that favor filtration are gonna be our capillary, glomerular capillary hydrostatic pressure. So the push force from the glomerulus out into the Bowman space. We also have the Bowman's capsule osmotic pressure. So the pull force from the Bowman's capsule pulling fluid from the glomerulus into the Bowman's capsule. So those two forces you're going to plug in as your filtration forces, and then that's going to be your first sum total. The other two forces are opposing filtration. So these are going to be your Bowman's capsule hydrostatic pressure. Again, think about where you're standing in the Bowman's capsule, and it's a hydrostatic force, so it's pushing. So that'll be pushing fluid back into the glomerulus, right? And then the other force that is opposing filtration is your glomerular capillary oncotic pressure. So you're standing in the glomerular capsule, the glomerulus, you're standing in the glomerulus and you're pulling fluid from the Bowman's capsule back into the glomerulus. That is going to be opposing filtration. So the forces that are favoring filtration are trying to move fluid from the glomerulus out into the Bowman's capsule. That is the direction of filtration. The forces that are opposing filtration are trying to move fluid in the opposite direction. So going back into the glomerulus from the Bowman's capsule. Okay. And so you're going to want to be able to plug those forces in and then calculate out and see what your overall value is. If it's positive, it is a filtration force. If if the filtration is bigger, it is also a filtration force. Okay. Someone is asking why increased resistance in the afferent arteriole leads to decreased glomerular capillary pressure. Excellent question, right? And so if we have more resistance here, we're clamping down on this vessel. Is more or less blood going to get into the glomerulus? Less blood, right? So if we're increasing resistance here, in other words, the smooth muscle that is lining the afferent arteriole is constricting or contracting, making the lumen here very small, less blood comes into the glomerulus. In other words, less blood can be filtered. In other words, we're going to decrease our glomerular capillary hydrostatic pressure, okay? Because we have less blood coming here, so less of it can be pushed out, and so we decrease this force, right? So that's a really important application of understanding the Starling forces. If I have less blood coming in here because I decrease the size of this vessel, I'm going to decrease the amount of fluid here, which is blood or plasma, and I'm going to decrease my hydrostatic force. Remember, the hydrostatic forces are owed to the presence of fluid in that space. The osmotic forces are owed to the presence of protein in that space, okay? Someone is saying, is the efferent arteriole carrying the filtered blood? So no, not at all. The efferent arteriole is carrying the blood that has not been filtered. Remember, we said that there's a filtration fraction. So all of the blood that ends up in the glomerulus doesn't get filtered. It's only about 20% that makes it across this membrane. The rest of it goes into the efferent arteriole, goes into the peritubular capillary, goes into the bassa recta, and then eventually into the interlobular vein, et cetera, it goes back to the vena cava, okay? So all of the blood that goes into the glomerulus does not get filtered. It goes back into the efferent arteriole. What is not filtered goes back into the efferent arteriole, whereas 20% of that eventually gets filtered across the glomerulus and makes its way into the tubular structures as that filtrate, okay? So the filtered bit ends up in the Bowman's space, the Bowman's capsule, and starts making its way through the peritubular space, through the proximal tubule, loop of Henle, etc., whereas the non-filtered bit ends up in the efferent arteriole. Okay. Any other questions pertaining to this region and the structures here? Any other questions here? So again, get comfortable with this idea of what these forces are being caused by. If I tell you that for some reason I have more protein here in my Bowman space, you should be connecting that to an oncotic force to start, but then the Bowman's capsule oncotic force. So that is going to be a force that is pulling fluid in this direction. And we know that there should not be proteins here because they cannot cross this membrane. But if theoretically in a question, I tell you there's a tear in the glomerulus, proteins end up in the Bowman's capsule, you should equate that to an increase in the Bowman's capsule osmotic force, okay? Likewise, if I tell you that there's more protein in the diet and more protein is in the glomerulus, you should equate that to more, an increase in the glomerular capsule, the glomerulus osmotic force, which is the presence of proteins here that's pulling fluid back in this direction, okay? All right. Other things to review, transport maximum. So let's go down here quickly and talk about that. So transport maximum versus, where is it? Transport maximum versus renal, renal threshold. And so here we talked about the facilitated carriers that are helping to reabsorb these substances, especially glucose. They have the ability to get saturated. And so they can become saturated at a specific rate, right? So if they're transporting 375 milligrams per minute, that is the rate at which they become fully saturated. They cannot go any faster, okay? And that is our transport maximum. On the other side of that same coin, we have the plasma concentration at which that saturation takes place, right? So if I have a lot of glucose in my blood, more of it will get filtered out. More of it will end up here in my proximal tubule to be reabsorbed across this epithelial cell. And so that plasma concentration at which that saturation occurs is my renal threshold. So transport maximum and renal threshold are two sides of the same phenomenon. We have saturation of the carriers that are responsible for reabsorption. And so saturation results in reabsorption no longer being able to occur. So the transport maximum is the rate that the carriers are moving at which they become saturated. Renal threshold is the plasma concentration of that solute at which my transporters become saturated. Okay, so renal threshold is a quantity, a blood level, so it's going to be milligrams per deciliter, whereas transport maximum is going to be a rate, it's going to be milligrams per minute, okay? But it's the exact same phenomenon. I can no longer reabsorb anymore because my carriers are fully saturated that help to transfer that substance across the basolateral surface and into the blood. All right, any questions on transport maximum or renal threshold, anything like that? So just looking at this graph here, when I get to transport maximum / renal threshold, I'm gonna stop reabsorbing. Reabsorption line flattens off. I'm still filtering it across the blood, it's still in the blood, it's still filtering it across the glomerulus, but now I'm going to start excreting it because I can no longer facilitate reabsorption because those carriers have been fully saturated. Okay. All right. And then another thing here in this packet was, where is it? Someone asked me to go over this question that was on your weekly quiz. So this question here having to do with clearance. And so this question asks, so the clearance of what substance, a natural byproduct of muscle metabolism, provides a non-invasive, and I went on to explain or describe what non-invasive means. So naturally found in the body, does not need to be exogenously introduced or injected into the veins, and is going to estimate GFR, our glomerular filtration rate. And so the meat of this question, the clues here are naturally occurring, so a natural byproduct of muscle metabolism, also non-invasive, right? We don't have to put it into the body, it's already in the body. And so we talked about two things that help to estimate GFR. Inulin, which is the more accurate estimator of GFR, but inulin is not naturally occurring in the body, it's actually occurring in plants. We have to put it into a vial, we have to inject it into our veins. And so that is going to be more invasive mechanism of estimating GFR. Creatinine, on the other hand, is a natural byproduct, right? When we break down muscle, when we break down proteins, we generate creatinine. And so creatinine is always in our body, always being cleared through the kidneys. And so that is a more non-invasive, a more natural, a more organic way of estimating GFR. You simply measure how much of that creatinine is coming out in the urine because it is already in the body. You don't have to exogenously put it into the body and then measure how it's coming out. Okay, so if I were to ask you, if I cleared all of this out the question, right, all of this out the question, and I simply said oh, sorry, here. I can't do this anymore. All right, here we go. So the clearance of what substance is the best estimate of GFR? The answer here would have been inulin, okay? If I put all of this stuff in, it's non-invasive, it's in the body, it's a natural byproduct or metabolic breakdown product, then creatinine is going to be the answer, okay? Just to explain that there a bit further. And then also make sure you have an understanding of the clearance. So how things are cleared versus this table that we went through. If the clearance is greater than GFR, secretion has taken place. If the clearance is less than GFR, the absorption has taken place. If the clearance is 0, then there is full reabsorption, such as glucose. So there's 100% reabsorption. If the clearance is the exact same as GFR, then no reabsorption or secretion has taken place, which is the case with inulin. All right. Okay. So this is why creatinine is not the best estimator, but it's the most, it's the least invasive. It's the best in terms of not interrupting the body's homeostasis. So we're not putting anything in, and it's being secreted slightly. So it's a little bit higher than GFR, but it is still a good predictor of GFR. Okay. Any other questions there? Any other questions there? Final 10 minutes, what I'm going to go through now is the distal convoluted tubule and just kind of organizing those hormones and then the JG cells. So let's go here. Actually, let's pull up the blank side. Okay, so we're looking at the distal convoluted tubule. And the collecting duct. Okay, this is where we have the regulated reabsorption. And really important, this is via the principal cells. These are the cells that line that part of the tubule. So we had 3 hormones that we talked about, and we're going to try to organize these hormones. I apologize for the scratchy lines here and make sense of them. I'm going to try to organize these hormones and make sense of them. I'm going to talk about where they are secreted. We'll talk about what they are, so the name of the hormone, We'll then talk about the stimulus for secretion, and then we'll talk about the mechanism. Really important how they bring about their action. Okay, so first of all, we have ADH. That's coming from the posterior pituitary. All right, the stimuli for ADH were twofold. So one was osmoreceptors, right? Remember the OVLT, so the osmoreceptors of the OVLT that detect an increase in plasma osmolarity. Right? In other words, very concentrated plasma, lots of solutes, little water. And so the goal of ADH is going to be to reabsorb more water and prevent the loss of that water. So that it would make sense that high plasma osmolarity would be a stimulus. The second stimulus for ADH was the decrease in baroreceptor stretch. So the decrease in baroreceptor stretch, which is, again, making sense. If we have less stretch, there's less volume, right, less fluid in the circulatory system. And so we're going to want to secrete ADH to bring more of that fluid back to help correct that decrease in baroreceptor stretch. The mechanism of ADH was to insert, so it's going to bind onto receptors in the basolateral surface of our principal cells, right? Remember, principal cells are what we're talking about here. And then it's going to insert aquaporin 2 channels in the apical surface. Okay, right. Let's talk about another hormone. So again, all of these start with A, so it's really important. I know students typically confuse these. The second hormone, aldosterone. All right. Aldosterone. So aldosterone secreted from the adrenal cortex. This is going to have a stimulus of an increase in plasma potassium, as well as a decrease in plasma sodium. Right, remember we said that the body was very sensitive to potassium levels. So if there was high potassium levels, we need to secrete more of that. And so that's going to be one stimulus for aldosterone. Mechanisms here were that we increase the sodium potassium pumps on the, on the basolateral surface of our cells, on the basolateral surface. We also insert more sodium, or let's say and, because it does both, and potassium leak channels on the apical surface. Right? If we insert more sodium, we can reabsorb sodium because that net direction is inward. If we insert more potassium leak channels, we secrete more potassium. The net direction, remember the sodium potassium pump is there. So if we're pumping potassium into the cell, the net direction of that potassium is out into the tubule, right? So by putting these leak channels in, we can reabsorb more sodium or we can secrete more potassium, all right? And that will correct our initial disturbances, losing potassium if it's too high, reclaiming or reabsorbing sodium, excuse me, if it's too low. The third hormone here is ANP. So ANP is our third hormone. This one is antagonistic to aldosterone. This is being secreted from the atrial wall. So the atria of the heart, atrial wall, ANP, atrial natriuretic peptide. And the stimuli for ANP secretion are stretch on the arterial wall. So if you have increased stretch on the arterial wall, we are going to increase our ANP secretion. Another stimulus was increased plasma volume, which is kind of synonymous with stretch on that arterial wall. So increase in plasma volume. In other words, we have too much sodium in the blood. We have too much fluid in the blood in the circulatory system. We secrete ANP, and that is going to block those leak channels and prevent the reabsorption of that sodium, which will help to correct that disturbance. So the mechanism of ANP is to block the sodium leak channels on the apical surface which essentially is preventing the reabsorption. So that's why these two are antagonistic. Aldosterone wants to bring more sodium back. ANP wants to lose more sodium into the urine by blocking those leak channels. Okay. All right. Any questions with these three hormones, their location, their mechanism? Let's see here. Okay, let's move on quickly to the JG complex. I think that's the final thing I want to review here. That shouldn't take very long. So the JG complex had three groups of cells. Juxtaglomerular complex. That had the macular densa cells. The macular densa cells were in the distal tubule. And they were responsible for detecting the flow rate, so how fast fluid is coming through that distal tubule. So the flow rate, as well as the sodium and chloride levels in the filtrate, right? So the flow rate and sodium chlorides in the filtrate. Second group of cells here were the JG cells. I'm also going to put paracrine factors here. Excuse me, so paracrine factors. And that is how macular densa can communicate to the second group of cells, which are the JG cells, juxtaglomerular cells, sometimes called the granular cells. These cells are found in the afferent arterial. Right, these are the ones that are lining that smooth muscle at the afferent arterial. They are detecting changes in GFR and mean arterial pressure, right? Remember, we talked about that auto-regulation. If there's a lot of fluid coming through, we're gonna clamp down. So if a lot of stretch is happening, we auto-regulate by clamping down, et cetera, et cetera. If there's a little bit of fluid, we can open up, et cetera. So auto-regulation. Yeah, this was myogenic auto-regulation, excuse me. Right, same idea here, that autoregulation or myogenic response. This is also going to secrete renin. So it's another kind of role here that the cell is playing, or sorry, kind of getting confused here. So this is going to secrete renin. And then the third cluster of cells was the mesangial cells. These are found in the vascular pole, so kind of tucked in between. And these are responsible for, again, GFR regulation and mean arterial pressure. These also are associated with paracrine factors. And so they can communicate to these cells as well, right? The paracrine factors helps them to tell the JG cells to constrict or to relax appropriately. Last thing I'm going to kind of squeeze in here on this slide in the last minute here are the stimuli for renin. So one is going to be sodium and chloride levels in the filtrate. So if there's low sodium or chloride. Two was going to be the afferent arterial pressure. Specifically, if that was low. And then three was going to be a decreased baroreceptor stretch. In general, anything that decreases mean arterial pressure will signal renin release. Okay, I think that's all